In this session, we are going to describe the structures within the cranial cavity, mainly the meninges, the the arachnoid and the dura, and the spaces between each of them. Then, mainly the subarachnoid space will describe it with its dilatation and the positions of these dilatations. Then the folds and the septa made by the dura mater, mainly the inner layer of the dura mater, and will end up by description of the dural sinuses, the venous dural sinuses. Now let us first identify the types of cranial cavity veins, those within the cavity or outside the cavity or within the bones of the skull, what call them the diploic veins. The first one is the emissary vein. Emissary vein is the vein which connects the outside of the skull with the inside of the skull. There are multiple and many veins, tiny veins, found within most of the foramina of the skull, which we have described in the skull. Those veins are important in equalizing the venous pressure on both sides of the skull and does not allow the intracranial pressure to be increased due to any venous reason or obstruction. That is the emissary veins. But the danger of those veins, it can transmit infection from outside the skull to inside the skull to the very sensitive structures within the cranial cavity, which is a very dangerous thing, especially from the face and also from the scalp and other regions which we'll come to know during the study of the head and neck. The second type of veins is what we call the diploic veins, and those veins drain the spongy part of the skull bones between the outer and the inner layer, and also is connected with the emissary veins outside and the veins inside the skull, but mainly to drain the skull bones. The third type is what we call it the meningeal vein, the one you can see on the outer surface of the dura. And this meningeal veins will drain mainly the outer layer of the dura and the other layers of the meninges. The last one is what we call it the cerebral vein. You found it on the outer surface of the brain here, but it drains all the parts of the brain, hemispheres and brainstem, cerebellum, all of them, and ends in the dural sinuses, the venous dural sinuses. These are the types of the veins dealt with in the skull, outside, inside, all within the bones. The first one, which is the spinal dura mater here, which is an extension from the cranial dura mater, that is the one here in light blue. It extends from the cranial cavity as continuation of the meningeal layer. In the skull, there are two layers, meningeal layer, the inner layer, and the outer layer, which is the endosteal layer. But the one in the spinal cord, around the spinal cord, is only the meningeal layer. There is no endosteal layer there. The main difference between both layers, the endosteal and the meningeal, is the nerve supply. The nerve supply of the endosteal is somatic, so it is very sensitive to pain and somatic pain stimuli. But the inner layer, like that of the spinal cord here, is not sensitive, being supplied by autonomic nerves. It encloses the spinal cord and, of course, above the brain, and end by at the coda equina, the lower level of the spinal cord at L2. But is attached above to the circumference of the foramen magnum. Below, it becomes thinner and invest the filum terminal. This one here, that is the filum terminal. That is the end of the dura at the level of the sacral 2, the second piece of sacrum. Then the filum terminal, which is supposed to be the termination of the bimatter, which end at the back of the coccyx, is invested by the dura mater. That is the filum terminal here. On each side of the spinal canal and also above along the cranial nerves within the cranial cavity, it extends with sheaths around the spinal nerves to end at the intervertebral canal. About the arachnoid matter, this is the second layer, which is deep to the dura, and this one is this one here, and a very thin, delicate tubular membrane loosely investing the spinal cord. Even it is said it's two layers, one toward the bia and one toward the arachnoid, the dura mater. But let us take it as a thin, delicate tubular membrane deep to the dura mater. The most important part above is continuous with the cerebral arachnoid, but the most important part of it is the space deep to it, which we are going to describe the subarachnoid space. When it comes to the bimatter, that's a very delicate membrane investing the spinal cord and the whole brain and brainstem and cerebellum. It goes in the cranial cavity with all the fissures, depressions, and elevations, sulci and gyri. And it's not easy to bake that bimatter, to separate the bimatter from the surface of the brain or the spinal cord. It gives the glistening appearance of the brain, which you look at it in the dissector room. This bimatter in the spinal cord, it extends around the spinal nerves between the anterior and the posterior roots to end in the intervertebral foramen. So it forms what we call a dentate ligament, something like that. It goes this way and come. So it is 21 pairs of triangular ligaments going from the side of the spinal cord to the intervertebral foramen. And this ligament helps in stabilizing the spinal cord in position. The filum terminal, as we said, is an extension of the bia below the conus medullaris, the lower part of the spinal cord, down to the back of coccyx, as we said. Now, this is about the subarachnoid space, but the other spaces are important also. We have a space outside the dura, which we call it epidural space, a space between the dura and the arachnoid, the subdural space, and the space which you can see here, the subarachnoid space deep to the arachnoid, between the arachnoid and the bia. The epidural space will be discussed in details when we come to the lumbar and sacral bones, and the contents will be dealt with mainly the semi-liquid fat and the roots of the lower part of the spinal cord, the coccygeal, the lumbar, and the sacral, and the most important part of this space is the internal venous plexus, vertebral plexus of veins. We'll discuss the importance of that plexus later. About the subdural, which is between the dura and the arachnoid, is a potential space. There is nothing in it. And any contents of any fluid, blood, pus, whatever, is pathogenic. The one we are talking about is the important one, which is the subarachnoid space. And this subarachnoid space is found between the bia and the arachnoid. And it contains, as we said, the cerebrospinal fluid. It has many spaces which call them cisterns. Cistern, a dilated part of the subarachnoid space. The most important one, the largest one, is what we call it the terminal cistern, the one which extends from the lumbar 2, where the spinal cord will terminate, to sacral 2. That is what we call it the terminal cistern. And this is the position for lumbar puncture. That's where the lumbar puncture will be found, will be done. That's how it looks like, the lumbar spinal puncture or taping. You are going to approach the subarachnoid space either between the third and fourth lumbar or between the fourth and fifth lumbar. This is the shows positions, mainly the lower one between 4 and 5. And this terminal cistern here, as you can see, is the one which can your needle reach easily without injuring the spinal cord, or even the nerves can pass between the nerves, and then you can do your lumbar puncture. There is other cisterns here. The largest one in the spinal, in the brain, in the cranial cavity, is what called cisterna magna, on the back of the medulla, between the medulla and the cerebellum. But there is what called chiasmatic at the optic chiasma. Another one, interpeduncular of the midbrain, interpeduncular cistern, and a pontine cistern. But the important one, which you can take sample of CSF, is that one which is the cerebello-medullary cistern, or cisterna magna. That's how you can approach to get cerebrospinal fluid from the cranial cavity. That is the cisterna magna. Here are different types of intracranial pressure. The first one is extradural, and usually due to fracture of the skull bones. It's extradural hemorrhage. The blood collects between the clavera and the preosteal, the endosteal layer. Or subdural, as we said, in the subdural space between the dura and the arachnoid, which is a potential space. Or subarachnoid, which could be in the subarachnoid space. These are the sites other than the intra-substance hemorrhages within the substance of the brain, but these are the intracranial, the most common intracranial hemorrhages. Now the nerve supply of the cerebral dura mater, the outer layer, we said it is supplied by somatic nerves. So here, that is the area which is supplied by the ophthalmic division of the trigeminal nerve. That is the area supplied by the maxillary division, V2. And that is the area supplied by the mandibular division. So these are the areas by the trigeminal nerve, on both sides, of course. The lower part of the posterior cranial fossa supplied by cervical nerves, carried by the hypoglossal nerve. The main nerves you can see there is the anterior marginal branch of the ethmoidal nerve, can be seen here very light. One which is the branch of meningeal branch of the maxillary, meningeal branches of the mandibular, the recurrent meningeal branch of the ophthalmic or the tentorial in relation to tentorium cerebelli. And lastly, on that you can see that is the tentorium cerebelli here. Its upper surface is supplied by those of the ophthalmic nerve, but the deep cranial fossa, posterior one, all of it is supplied by the cervical second and third with the hypoglossal nerve. That is the nerve supply of the dura outer layer. Now the dural septa, there are 5 septa, but mainly the main one are 2. The cerebral falx or the falx cerebri, this one here, and the tentorium cerebelli, this one here. These are the 2 main folds which we have to study them in details. And then that is the falx cerebelli, you can see it on both sides here. And that is what we call it the diaphragma cilia here, covering the pituitary gland and in the pituitary fossa, which is this one here. And lastly, there's a small fold which called cavum trigeminal, which covers the trigeminal ganglia. Now, that is the relation of these folds together. That is the falx cerebri on the upper surface of the tentorium, and this is the falx cerebelli on the under surface of, that is the tentorium cerebelli here. That is the falx cerebelli on the under surface of the tentorium, and the falx cerebri on the upper surface. As I said, these are the 2 main. You can add the falx cerebelli. We'll see the diaphragma cilia covering the cilia turcica or the pituitary fossa. Let's start by the falx cerebri, one of the important ones. That is the one here. That is the falx cerebri. It's a sickle-shaped, having an apex, a base, a superior border, and inferior border. That is the borders of that falx. It's very strong, concentric in shape, as we said, lie in the distal plane, occupying longitudinal fissure between the 2 cerebral hemispheres. So the 2 cerebral hemispheres are found on each side of this falx. The function of this falx cerebri is very, very important, as it's attached to the vault of the skull, as you can see here on the undersurface of the vault of the skull, to the sides of the groove of the superior sagittal sinus. You can imagine the 2 hemispheres on each side lying on the upper surface of the tentorium cerebelli on each side. So that falx cerebri is carrying the cerebral hemisphere weight, hanging them to the undersurface of the vault of the skull, preventing that weight of the hemispheres to press on the structures within the posterior cranial fossa. I hope this is clear. The 2 cerebral hemispheres are resting on the 2 sides of the tentorium cerebelli, and that cerebral hemisphere by itself is hanging to the undersurface of the vault of the skull. So it prevents pressure on the structures of the posterior cranial fossa, mainly the medulla oblongata and the cerebellum, mainly the tonsils of cerebellum on each side of the medulla. Increased intracranial pressure will lead to what we call it herniation of the medulla. The 2 tonsils also will be herniated, blocking the foramen magnum, which lead to more increase and increase of the secreted cerebrospinal fluid. And this will press on the cardiac and respiratory centers found in the medulla and could be a deadly situation. That is the importance of that falx cerebri. It carries the whole thing up, being attached to the upper surface of the tentorium cerebelli, carrying that tentorium with the weight of the hemispheres to the vault of the skull. Now, it has an apex, as we said, fixed to the cresta galli of the ethmoid. It has a base, that is the base here, attached to the upper surface of the tentorium cerebelli, where the straight sinus will be passing or running between the base and the tentorium cerebelli. That's where the vein will be found here. The lower edge arches over the corpus callosum here. So these are the lower edge here, arching over the corpus callosum. It is a free border. This one is an attached border, and the base also is attached border. This one is a free border. Its convex upper border, as we said, is attached to the internal cranial surface on each side of the midline, as far as back at the interoccipital protuberance. The superior sagittal sinus lies in that cranial groove. The tentorium cerebelli, which is the shape of a tent or a single ball tent being raised from the middle by the falx cerebri, it has two sliding sides on each side. That is the one here. It lies between the cerebellum and the occipital lobes of the cerebral spheres, dividing the cranial cavity here into two regions. Supratentorial compartment, which contains the forebrain, and infratentorial part, which contains the hindbrain, the cerebellum and the brainstem. The borders are the free border, and that free border surrounds or first separates the dorsum silli of the sphenoid bone by a large curved hiatus which contains the midbrain, and the anterior part of the superior aspect of the cerebellar vermis, but mainly the midbrain in that notch. We'll look at that notch in another view. The anterior end of that free border will extend to the anterior clinoid process to be attached there after crossing the attached border. The attached border, or you can call it the posterior border, that is the one here, it's convex and is attached posterior to the lips of the transverse sulcus posteriorly of the occipital bone and to the posterior inferior angles of the parietal bones, between the occipital and the parietal bones. And there is the transverse sinus. Laterally for that attached, so it has a lateral here and posterior here. It is a sort, we'll see it in the next diagram. They attach it to the superior border of the vitreous part of temporal bone, as you can see here. That is the superior border of vitreous bone here, where that attached border is attached to the vitreous part, the posterior part to the occipital and the parietal. And along this one will be the superior parietal sinuses. The anterior end is attached to the posterior clinoid process being crossed by the free border. That crossing of the anterior or the free and attached border is a landmark for the exit of cranial nerves. At the point of crossing, there will be the trochlear nerve piercing the dura coming out from the brainstem. Anterior to this discussion, the decussation, is the oculomotor nerve. We'll see that in the next slide. And behind is the trigeminal. That's how it looks like here. That is the relation of the nerves to the tentorium cerebelli. That is the free border here, with the midbrain in the incisura or the notch here. The anterior border will end in the anterior clinoid process, while the attached border, this part for the superior petrosal sinus, this part for the transverse sinus, here will be the straight sinus over here. At the point of discussion, the trochlear nerve will be coming out. Anteriorly will be the oculomotor. Posteriorly and a bit lower will be the trigeminal nerve, the largest cranial nerve, the thickest cranial nerve. That is the dural septa here, which we have that diagram before. The falx cerebelli, this one here, is a small midline fold between the two hemispheres of the cerebellum. It bulges in the posterior cerebral notch on the back of the cerebellum. Along its free border is the occipital sinus, and being very small, it could be along the attached border also, to end in the straight sinus over here. That's the falx cerebelli. The base is direct upwards and attached to the posterior part of the inferior surface of the tentorium cerebelli, as you can see here. The posterior margin contain, which now is the attached border, contain the occipital sinus and attached to the internal occipital protuberance. That is the diaphragma cilia here, seen from above. You can see that is the boundaries of the cilia turcica. That is the pituitary gland with the infundibulum bulging through an opening within the diaphragma cilia. That is the diaphragma cilia here. A small cylindrical part of the dura. Form the roof of the cilia turcica, roofing the pituitary and completely covering the pituitary gland. The infundibulum, as you said, and the pituitary stalk pass through an opening or a hole in the upper surface. The cavum trigeminal, which we cannot see over here, it is almost will be around here, around this part, almost this part here, at the apex of the vitreous part of the temporal bone, where the trigeminal ganglia is lying in that fossa. It covers the trigeminal ganglia. That is the cavum trigeminal. Now the dural sinuses, the dural venous sinuses, what would like to know about it? First, they are divided into bird and single. The bird are the sphenoparietal or sphenoorbital along the posterior border of the lesser wing of the sphenoid. Two cavernous sinuses on each side of the body of the sphenoid here, that is the cavernous sinus here and here, which will open into the superior and inferior petrosal. I mean, the cavernous sinus will end up into superior and inferior petrosal sinuses. So that is the superior petrosal, which will end up by uniting with the transverse sinus to form the sigmoid sinus, while the inferior petrosal sinus will go out the skull cavity to join the internal jugular vein below the skull. So the superior petrosal, as I said, will join the transverse to form the sigmoid sinus. That is the bird sinuses. Here also we can see the ophthalmic veins of the orbit. We can see the basilar sinuses at the clavus of the skull. It's very good demonstrated here. And this is the occipital sinus along the internal occipital crest here, the attached border of the falx cerebelli. And this is the intercavernous sinuses, anterior and posterior one, connecting the two cavernous. The intercavernous, the basilar sinuses are single and not paired, plus the occipital. So the occipital, the basilar, and the intercavernous are of the single ones, right? But they are clear in that slide. Here, the superior sagittal is very clear. The inferior sagittal sinus, the straight sinus at the base of the falx cerebri, at the upper surface of the tentorium cerebelli, the occipital, which we have seen here, that is the occipital which join the straight, the basilar over the baso-occipital, it is more in the previous slide, more clear I mean, and the occipital, the intercavernous also are more clear in the previous slide. So these are the single. Here we can see also the great cerebral vein which join the straight sinus, with the superior with the inferior, the inferior sagittal with the great cerebral, they form the straight sinus or end in the straight sinus. That is whole dural venous sinuses. That is an X-ray showing those sinuses again. That is the sigmoid sinus here ending in the internal jugular. That is the internal jugular sigmoid sinus. The same thing here. That is the sigmoid entering in the internal jugular. That is the superior sagittal here and here. You can see the superior sagittal is going to the right side here. In certain cases, it will go to the left, or it will unite with the straight to form venous confluence. That's how it goes. We'll talk about it now. This is the internal jugular here and here. This is the inferior sagittal sinus. And this is the transverse sinus here and here. That's a good X-ray view of those sinuses. Here, a summary of this dural sinuses. The blood flow within the dural sinuses. It will go backward from the front to the back to join the transverse sinuses, as I said. The superior sagittal sinus will join the right. In this case, you will find the right internal jugular vein larger than the left one. You can see the difference of the size. Or it will join the left. This is around 10 to 15%. In this case, the left will be larger than the right. Or two transverse sinuses will unite, where the superior sagittal, inferior sagittal, and the straight sinus will end in a venous confluence, and then equally distributed to both transverse sinuses. In this case, both internal jugular will be of the same size. So it goes backward in this direction, then through the transverse sinus, it will go forward, and then downwards through the internal jugular veins. The main difference between the sinuses and the veins are the sinus wall has no muscle layer, so no corrugation could be in the inner surface of the sinus. The cavity is smoother of the sinus compared with the vein, so it decreases the possibility of blood clotting within the cranial cavity. And also, having no muscle layer, cutting of the sinus, the muscle fibers which are not there, if in the vein, it can form increase the gap of this by muscle contraction, can increase the tone of the muscle, will increase the gap of bleeding, but this will not happen in the dural sinuses. It has no valves in the dural sinus, so allows the blood to go from any direction, from the front to the back or the back to the front, in case of a localized increase in the intracranial pressure for a tumor, for a blockage of a vein or whatever. That is the difference between a sinus and a vein. Now, we can see here the diploic veins opening in, that is the superior sagittal sinus here. And this is the emissary vein reaching again what we can call it, that is the dura mater here, which divides into two layers, as you can see here. And this is the subarachnoid space over here. That is the subarachnoid space. That is the biomatter close to the surface of the brain. That is the arachnoid here, close to the inner layer of the dura mater here. And that is the cerebral cortex here. And this is the falx cerebri, which is formed by the inner layer of the dura mater, as you can see here. That is the meningeal veins, which we talk about on the outer surface of the dura mater. And here is the cerebral veins here, which can communicate with this or end in these sinuses. That's the venous lacunae on each side of the superior sagittal sinus, which can be seen on the outer surface of the brain also, of the dura, I'm sorry. And the arachnoid granulations here and here. This is the superior sagittal sinus, as we said, this one, which is this one on the other side, the whole from the anterior the part at the frontal crest down to the internal occipital protuberance. And this is the emissary vein which we have seen. Now, the cavernous sinus has some relations which is very important to know. It has contents to start with. The third nerve, the oculomotor, in the lateral wall, with the trochlear nerve below it, the ophthalmic division of trigeminal, and the maxillary division of the trigeminal, the four of them in the lateral wall. The sixth of the trochlear, the abduced nerve, is found in the roof, just lateral to the internal carotid artery. The pituitary gland is on the medial side. The internal carotid artery is above and in the floor. It goes in the floor first, then upwards, piercing the sinus to go backward above the sinus. So it has two relations with the cavernous sinus. The sphenoidal air sinuses are below and medial to it. The temporal lobes are lateral, especially the uncus, the part of the temporal lobe.